Nanoparticles are increasingly used in industry and medicine due to their unique physico-chemical properties. However, concerns for human health associated with prolonged nanoparticle exposure persist. These risks can be quantified by studying the behavior of nanoparticles inside cells and the induced metabolic responses of cells to nanoparticles. This is partially due to the lack of methods that allow the detection and quantification of internalized nanoparticles in a single cell. While numerous analytical tools, including microscopy and mass spectrometry, can be used to estimate the cellular uptake of nanoparticles, these only provide qualitative information at a macroscopic level. On the contrary, in situ methods based on atomic spectroscopy can reduce the quantity of imaging artifacts that arise from sample preparation and can yield direct observations of nanoparticles. To take advantage of this, we present a correlative approach which allows us to study nanoparticles either in their native state or labeled with a fluorescent tag. Here, we describe a method based on the combination of fluorescence microscopy and nuclear microprobe analysis. This provides images of the cellular density, chemical element spatial distribution, and the amount of nanoparticles per cells. As a demonstration, we show results from cells exposed to titanium dioxide nanoparticles over 24 hours using both fluorescence microscopy and nuclear microprobe analysis.
For this protocol, a custom sample holder made with the polymer PEEK is needed. It must be suitable for cell culturing and in vitro observations. Cover the holder with 2-micron thick polycarbonate foil. The polycarbonate foil is glued to the